welcome you all to the second session of Molecular Diagnostics and Genomics track. The next speaker that I have is Xenia Nazarova. She is a senior research scientist, DNA technology in Moscow, Russia, and an immunologist with extensive experience in molecular diagnostics, infectious diseases, and skin microbiome research. She holds a master's degree in molecular biology and immunology from the Moscow State University of Veterinary Medicine and Biochemistry. Currently, as a senior research fellow in DNA technology and a researcher at the State Scientific Center Immunology Institute, she specializes in the development of real-time PCR diagnostic systems, nucleic acid extraction technologies, and regulatory documentation in compliance with ISO 13485 standards. Her research intersects include dermatological infections, antimicrobial diagnostics, and the human skin microbiome. She's an active contributor to the scientific community, having authored multiple peer-reviewed publications and presented at numerous national and international conferences. Her work bridges applied diagnostics and clinical research with a strong focus on translating advanced molecular technologies into practical healthcare solutions. She's going to talk to us about molecular diagnostics for infections of the skin, hair, and nails. Please join me in welcoming her.
Good afternoon, ladies and gentlemen. Allow me to introduce the topic of my presentation. It's molecular diagnostics for infection of the skin, hair, and nails. Human skin is a complex ecosystem with a diverse microbial population. This microbiome includes bacteria, fungi, viruses, and mites. A healthy normal microbiome provides a natural protection against external factors. Generally, the greater the microbiome's diversity, the stronger this protective effect. A healthy normal microbiome has a great microbiome diversity. However, the disruption of this homeostasis can occur due to damage to the skin barrier, shifts in skin pH, immunodeficiency states, and prolonged antibiotic use, which can reduce microbiome diversity and lead to infection. Some key drivers of this issue include, first of all, environmental and social factors. These include climate change, urbanization, and aging population. The second one is medical therapeutic factors, prolonged use of antibiotic and antifungal therapy, the growing the challenges of antimicrobial resistance, and increase in the immunocompromised patient population. And also the medical legacy of COVID-19 pandemic. And the third one is diagnostic challenges. That can be gaps in diagnostics, a lack of adequate comprehensive diagnostic system. Both bacterial and fungal infections are rising annually. Based on research and epidemiological projection, by the year 2050, we can expect a significant increase. For example, for bacterial infection are forecast to rise by over 37%, but for bacterial infection are forecast by rise by over 50%, and it's really great. There are geographic hotspots with higher risk. These include Western and Eastern Africa, and also South Asia. Regarding bacterial infections of the skin and hair, several primary types are distinguished. These include folliculitis, furuncles, impetigo, and hidradenitis. Fungal infections of the skin and hair can be categorized into several groups also. This can include dermatophytosis, keratomycosis, and candidiasis. Based on International Classification of Diseases, 11th code, we can categorize infection on fungal and bacterial etiology under the following codes, which are presented on the slide. Currently, in the practice of diagnosing bacterial and fungal infections, several key methods can be distinguished. First one, it's microscopy. It's really fast method, but it has a great limitation. For example, while it's fast, it doesn't tell us which exactly pathogen we are dealing with, but it's really important for choosing right treatment. The next one is culture-based method. While it's accurate, but it's a lengthy process, and it takes from 2 to 6 weeks for delivering results, especially for fungal infection. And it's really unacceptable in a modern clinical settings, and sometimes a patient lives without any diagnosis, and treatment is based often only on a clinical judgment. And the last one, it's molecular method, especially polymerase chain reaction. That method solves all problems. It's fast, it's accurate, and it has a great sensitivity and specificity as well. When we compare different methods with different parameters, PCR surpassed others' methods across all parameters. This includes sensitivity, specificity, species identification, analysis time. Microscopy surpassed PCR, but the result delivery will be the same as for microscopy and PCR. But why it's important to diagnose etiological agent? Because fungal skin infection often presents with bacterial infection as well, and bacterial species affect therapy choice. Different bacteria require different antibiotics, and incorrect therapy can lead to worsen dysbiosis. Antifungal choice depends on species identification as well. Different type of dermatophytes also demand different antifungal therapy, also different species of mold and yeast. Treatment can be topical and systemic. Topical therapy is typically applied when the infected area is not extensive, and systemic therapy is used then there is no improvement after topical treatment, and the infection covers a large or widespread area. The infection is particularly deep, salted, or severe. Treatment for fungal infections follow a similar principle in choosing the route of administration. Topical antifungal therapy is used when the area of spread is limited, not so large. Systemic antifungal therapy is indicated when large body surfaces are affected or not infection extends. Antifungal therapy is primarily based on the specific genus and species. Terbinafine, for example, is a first-line drug for treating infection caused by trichophyton species. Griseofulvine, effective against 2 microsporum species, and fluconazole is effective against Candida species. Wherefore, they first need to determine at least the genus to which a particular fungus belongs. Unfortunately, microscopy cannot identify the species. It can only determine the presence or absence of fungal elements like HIPAA in the sample. It's a simple and fast method. It's like you will place on your sample on the glass slide and apply with potassium hydroxide, and after that you will examine your sample under a microscope to visualize your HIPAA and spores. That's why there is no identification at all. We just can say there is a fungal element or no. And disadvantages, it's subjective. Laboratory assistant has to have a great experience in this field to differentiate what is it, fungal elements or artifactals. And also it has low sensitivity and specificity. That's why microscopy not a method of our choice in identification infections. This slide presents 2 cases of atopic dermatitis. The first case is uncomplicated by infection. PCR analysis revealed no significant overgrowth of bacteria or fungi and no absolute pathogens. were detected, such as dermatophytes. Therefore, the therapy for this case will correspond to standard atopic dermatitis treatment without the additional of antifungal agents or antibiotics. In the second case, we detected an overall elevated bacterial load, a specific overgrowth of Staphylococcus, and specific overgrowth of Malassezia. And according the treatment will be supplemented with targeted topical anti-staphylococcal therapy, and according the treatment, of course, with atopic dermatitis treatment, and antifungal therapy to address the Malassezia overgrowth. While the culture was positive in both cases for Staphylococcus aureus, its mere presence is not diagnostic, as Staphylococcus aureus can colonize the healthy skin without atopic dermatitis or others dermatosis at all. And only a quantitative PCR results is essential here to guide therapeutic decision making. Based on this quantitative data, antimicrobial therapy would be indicated in one case, but not in other. The next two cases present of seborrheic dermatitis and scalp mycosis. In the first case, no elevated levels of bacteria or fungi were detected, and no pathogenic fungi were found. Therefore, the therapy will be conducted according to standard treatment for seborrheic dermatitis. In the second case, with scalp mycosis is presented. In the first case, seborrheic dermatitis was diagnosed by the physician immediately. However, in the second case, the patient went approximately 6 months without a correct diagnosis because the culture result was negative. It was only with help of PCR analysis that we were able to detect the dermatophyte Trichophyton tonsurans. After that, the therapy was correct. Our company produced and developed some kits for diagnostic different infections of skin and hair. This panel, trichoderm screen, includes bacterial targets, fungal targets, especially absolute pathogens, dermatophytes, absolute pathogens, Trichophyton species with identification, Trichophyton rubrum, Mentographitis indotina, it's a resistant strain which we will discuss a little bit later. Microsporum canis and Epidermophyton flacosum, the main and only species in the Epidermophyton genus. And the rarer case is Nanesia species. It's also a dermatophyte, it's a new one. And the next part is yeast-like fungi, include Candida species with identification Candida albicans, Trichosporum species, and Malassezia species. The second case, kit, Trichophyton species, Microsporum species, is a kit for quantitative analyzing and controlling our therapy with predictable mycosis. And the last one, mycosis-resistant Trichophyton, for detecting Trichophyton species as genus at all. And identification Trichophyton Mentographitis indotina is a resistant strain. And detection, a single mutation which can be resistant to the first-line drug, terbinafine. The next part of my presentation is nail infections. Onychodystrophy may be infectional etiology and non-infectional, because this includes psoriasis, onycholysis, paronychia, and onychomycosis is the most common infectious nail pathology. The global prevalence of onychomycosis is average 5.5%. And risk factors include a family history and tinea pedis or mycosis of feet, tinea menum or mycosis of hands, wearing tight or close footwear, wearing gloves, and nail trauma. Laboratory confirmation is necessary for the true etiological diagnostic and proper therapy selection. As I said, onychomycosis onychodystrophy can be infectional etiology and caused by dermatophytes. It's the most common infection of all onychomycosis, and non-dermatophytes such as molds and yeast. Also, it can be bacterial etiology like Pseudomonas aeruginosa or green nail syndrome. But we should remember that only dermatophytes are true etiological agents. Yeast and molds may be skin contaminants. That's why quantitative analysis is really important for understanding our reality, our real clinical picture, and differentiate where is it, what is it? Is it normal colonization or it's inflammation caused by infection? And also, with different etiological agents can be together in the nail. Dermatophytes are pathogens with active invasion mechanism. Keratinized eczema break down keratin and penetrate full thickness of nail plate. Due to their pronounced keratolytic activity, dermatophytes penetrate the entire thickness of the nail plate, forming a network between corneocytes and inside the corneocytes. And keratolytic activity from dermatophytes enables superinfection by mold and yeast, which can be presented on the nail before. Onychodystrophy, if it's onychomycosis, it will be one therapy, systemic therapies, if it's dermatophytes. It's a long treatment course from 6 to 12 months. Sometimes it can be 2 years for treating. It depends of nail grown. And non-dermatophytes, we use topical therapy like cream or ointments. In this comparative study, authors compare different methods for different biomaterials, such as skin, hair, and nails. And PCR method was more sensitive than culture and microscopy method as well. And culture method for both biomaterials, skin and hair, was more sensitive than microscopy. But for nails samples, microscopy surpassed culture method. And PCR surpassed culture method in 24 times greater. There is a culture limitation. As I said, it's low sensitivity and time-consuming. Culture grow, especially for fungus, really long time. It depends on species. For example, Microsporum canis is growing like 7 days, and Trichophyton verrucosum will take 35 days, and it's really long time. And also there can be contamination. Grown microorganisms may be contaminants and non-pathogens. And there is a problem for mixed infection because traditional culture method detects only fast-growing microorganisms in the culture medium. We also decided to compare the difference in sensitivity between the different methods. For this, we used pure culture of Trichophyton interdigitale. We took this culture at a concentration of 10 to the power of 6 colony forming units per milliliter and performed a serial dilution down to 10 to the power of 1 colony forming units per milliliter. From each dilution, we took 100 microliters aliquot for PCR analysis and 100 microliter aliquot for culture-based method. Using PCR, the pathogen was detected in solution corresponding to 10 to the power of 2 colony-formal units per milliliter, and using the culture method, growth was only achieved from the dilution of 10 to the power of 3 colony-formal units. However, it's crucial to note that these were pure culture, and this situation becomes completely complex with real biological material such as nail, where fungal mycelium is deeply embedded with nail keratin, making it even harder for traditional culture methods to recover the organism compared to detection offered by PCR method. For example, when we compare the two methods for Trichophyton rubrum, the culture method only detects the fungus in sample where its concentration was above 10 to the power of 5 DNA copies per milliliter, as quantified by PCR. For Trichophyton interdigitale, this threshold was also around 10 to the power of 6 DNA copies per milliliter. The culture method failed to detect pathogenic fungi in nails where the fungal concentration was low. This was only possible using PCR analysis. In contrast, the PCR method detected both low copy and high copy samples far below this concentration threshold. Dermatophytes are located inside the nail throughout the entire thickness of nail plate. During microsporum, we use potassium hydroxide, and this dissolves the keratin matrix of nail, making fungal elements accessible for visualization. Under natural condition, Trichophyton rubrum and Trichophyton mentogrophytis complex grow with a limited titer of 10 to the power of 3 coloniformal units per milliliter. During culture method, the application of potassium hydroxide is impossible because we need to grow our live culture. Wherefore, a significant portion of the fungals remains trapped, or we can say locked inside the tissue matrix, and is unavailable for contact with culture medium. And our company also has a kit for detecting the main group of infection, of nails infection. This includes as bacteria and fungus infection. As bacteria, Pseudomonas aeruginosa is the main and only common pathogen known as the green nail syndrome. And for dermatophytes, we include targets like pan-dermatophytes. It's a marker for detecting the DNA of dermatophytes in the sample at all. Its clinical meaning, because Trichophyton species is the main group of dermatophytes caused to onychomycosis. And the next one is mold and yeast. It can be contaminants, and that's why we decided to use quantitative analysis of all pathogens into this screening kit. This slide presents two more cases. In the first case, it's an onychodystrophy with a mixed infection. In this patient, we detected Trichophyton rubrum, Fusarium species, and Pseudomonas aeruginosa. The nail dystrophy sero with hyperkeratosis confirmed by PCR. Unfortunately, the culture method failed to detect Trichophyton. In this case, it only showed presence of Fusarium species in the sample. Consequently, the therapy will need to include systemic antifungal therapy, topical therapy, and targeted therapy against Pseudomonas aeruginosa. In the second case, only Dermatophyton rubrum was detected. This is a typical dermatophyte and the sole cause of nail infection, which no bacterial co-infection. However, in this case, confirmation was only possible with PCR because culture method failed to identify the pathogens. Given the extensive area of nail involvement, systemic therapy was applied. And what is the aim and particulars of laboratory diagnostics? It's like summarizing. To confirm or rule out infections origin of nail lesion, to confirm or rule out dermatophyte infections, and to confirm or rule out co-infections of dermatophytes with non-dermatophytes molds and candida. And the last part of my presentation is Trichophyton resistant. This resistance is now a globally widespread phenomenon reported in over 40 countries. The first the first report about the resistance was from India in 2018, and now the primary method for detecting the resistance is molecular diagnostic because the strain is really challenging to grow that strain from biomaterial. The antifungal action of terbinafine targets a key enzyme in fungal cell membrane synthesis pathway squalene epoxidase. By inhibiting this enzyme, terbinafine effectively blocks the biosynthesis of ergosterol, an essential component of the fungal cell membrane. In response, resistant strain of dermatophytes develop specific mutations in the correspondence of ARG1 gene. These mutations alter the enzyme structure, making it less sensitive to terbinophine's inhibitory effects while allowing it to retain partial functionality. This compromised interaction leads to a dual cellular consensus. The toxic accumulation of the substrate, squalene, and disrupted ergosterol production pathway. This biochemical adaptation ultimately confers clinical resistance, rendering terbinafine therapy ineffective against such a strain and posing a significant challenge in the treatment of dermatophytosis. And this is a clinical case of resistant female, and her lesion was after trip to Indonesia in December 2023. The treatment was based only in a clinical judgment because doctor, based on her experience and expect mycosis because it's clinically visible that it's mycosis, and prescribed to a patient a terbinafine but without any response. And after in May 2024, we got a scalp scraping from skin of patient and analyzed it with PCR and detected trichophyton indatina strain, its resistant strain, with detected mutation in ARG1 gene 436 mutation. And after that, doctor changed therapy to itraconazole, and by winter 2024, it's complete clinical remission. And I want to summarize my presentation and say that PCR method has a great sensitivity and specificity. There is a species identification, it's really fast, and this is a key to success in mixed infection and resistant infection as well, and there is an objective result. Thank you for your attention.